Good morning, students. This is Rama, JL in Botany, and welcome to webinar 5 session. Yesterday, we discussed about the importance of pigments in photosynthesis. Today, we are going to discuss the mechanism of photosynthesis. Photosynthesis is a oxidative and reduction process in which H2O molecules are oxidized and CO2 molecules are reduced by utilizing assimilatory power. Here, the question is, what is assimilatory power? Assimilatory power means energy form that is in the form of ATP and NADPH2, which are synthesized during the light phase of photosynthesis. So, here the important point is this photosynthesis is an oxidative reduction process. And if we go through this photosynthesis takes place in two phases. First one is light phase, second one is dark phase. Light phase. It is a phase which takes place in the presence of sunlight. And in this light phase, oxidation of H2O takes place based on photochemical reaction. And the next is, it is affected by quality and quantity of light, but not by temperature. It means this phase is mainly influenced by light only, but not temperature. And third important point during light phase is, it takes place in grana portion of a chloroplast. Yesterday, the people were discussed about the structure of chloroplast, in which the stacks of thylakoids are called as grana. That means in grana, we can find so many of thylakoids. Thylakoid is a plate-like structure which bears pigment, chlorophyll pigment. So, in grana part, usually light phase takes place. Then next important phase is dark phase. In photosynthesis, it will be completed in two phases. The first one is light phase, second one is dark phase. And now just only we have seen this light phase and its characteristic features. That means during light phase, only oxidation of H2O based on photochemical reaction. The next is, it is influenced by light only, but not by temperature. And light phase takes place in grana part only. And moreover, during this light phase, we can say this one. What is that? Assimilatory power. That means 3 ATPs and 1 NADPH2 will be synthesized during this light phase. So, 4 important points in light phase. And about dark phase. What about the dark phase? Dark phase is a reduction of CO2 based on biochemical reactions, whereas light is a photochemical based reaction based phase. But here dark phase is biochemical reaction based phase is dark phase, in which CO2 is reduced. Then second point, this dark phase is influenced by temperature, but not by light. That means it is opposite to the light phase. And third one is, during dark phase, what is the end product? During the dark phase, end product is star. And to regenerate it, RUBP. So finally, end products are starch, then RUBP. And all these changes or all these reactions will be take place in soma part of chloroplast. That means a stroma is a jelly-like or it's a gelatinous substance which presents inside the membranes of the chloroplast. So this is about the briefing of phases in photosynthesis. So, under mechanism of photosynthesis, you have to remember two phases. That means the photosynthesis process will be completed in two phases. One is light phase, the other one is dark phase. During the light phase, once again, I'm repeating, this light phase is the first phase of photosynthesis, which takes place in grana part of the chloroplast. Grana. What is grana? It's a stack of thylakoids. What is thylakoid? Thylakoid is a plate-like structure which helps in storing pigments. So, light reaction requires 4 factors. Out of 4, chlorophyll pigment is one of the 4 factors. So, that chlorophyll pigment will be stored in thylakoid like a plate-like structure. And this plate-like thylakoids are arranged in groups. Each group is called as grana. So in that grana portion, the light reaction takes place. And this light reaction is influenced by light only, but not by temperature. And this light phase is purely, it's an oxidative process. And during this oxidation, H2O is oxidized based on photochemical reaction. And the other second phase is dark phase, which takes place in stroma part of the chloroplast. And this dark phase is purely temperature-based reactions, but not by light-based. That means this dark phase is influenced by temperature only, not by light. And this dark phase is CO2 based. That means a reduction of CO2 takes place during the dark phase, which is a biochemical reaction. So this is about the introduction of a photosynthesis process. Now, let us start the light reaction in detail. Right. Light reaction itself is indicating the need of light, and the source of the light is sunlight. So sun is the source of light. In the presence of sunlight, it takes place, and assimilatory power is synthesized during this light phase. And what is assimilatory power? 3 ATP plus 1 NADPH is togetherly called as assimilatory power. And this light phase takes place in grana portion or grana thylakoids of chloroplast. Then this light reaction is influenced by the light. Yes, the rate of photosynthesis was more when the chloroplasts were exposed to intermittent light. That means chloroplasts, they absorb the sunlight from the sun in the form of photons. And continuous flow of photons will not help in increase of photosynthesis or increase of rate of photosynthesis. Instead of continuous flow of photons, intermediate light, that means the breaks between the transmission of light, there should be splittings or there should be some interruptions, and that interrupted light that helps in increasing the rate of photosynthesis. What is the reason behind this? That means a continuous flow of photons will not help in increasing the rate of photosynthesis, whereas intermediate light means splitted or flow of this photons will be splitted into or will be will be what disturbed or they phase-wise flow of photons helps in increasing the rate of photosynthesis. And reason behind that is non-accumulation of ATP and NADPH2. That means continuous flow helps in continuous flow of utilization of assimilatory power, and that results the normal increase of photosynthesis, but not extreme increase rate of photosynthesis. That means this intermittent light helps in storing the assimilatory power in leaf, which helps in increasing the rate of photosynthesis. So here we should understand one point, that continuous flow of photons will not help in increasing the rate of photosynthesis, but intermittent light helps in increasing the rate of photosynthesis. That is the most important point. The next important steps in light reaction. First one is photochemical phase. Second is light absorption. Third is water splitting or photolysis of water. And fourth is oxygen release. And fifth one is formation of high chemical intermediates like ATP and NADPH. All these are the important steps which are discussed one after the other. Now, next, coming to the next part. Yes, during the light phase, important events of light reaction are, number one is photolysis of water, and number two is photophosphorylation. First, let us discuss about the photolysis of water. What is photolysis? Photo means light. Lysis means splitting. Photo means here in the definition, definition itself indicates what? Means photo means light. Lysis means splitting. So here in the presence of light, which molecule will be splitted into different particles? That means one H2O molecule, one water molecule splits into H plus proton and electrons and oxygen. First, initially, photolysis, H2O splits into H plus and OH minus. And OH minus furtherly splits into oxygen and protons. This oxygen will be released out as a byproduct, and the protons, whatever protons are released by this photolysis are transferred to reduce NADP plus into NADPH and H. So that is the importance of photolysis. So in photolysis, one water molecule splits into one proton and an electron and an oxygen. So all these changes happens only by the presence of light. So it is called as photolysis. That means splitting of water molecule into protons, electrons, and the oxygen is called as photolysis. And it is by light activated chlorophyll molecule. All these changes will be happened by the light activated chlorophyll molecule. That's why it is called as photolysis. In our single line, it is splitting of water molecule into H plus and OH minus in the presence of sunlight is called as photolysis. Why it is required during light reaction? There is a reason for this photolysis that will be explained in further slides or during the light reaction process. So it is essential for light reaction. This photolysis of water is the most important change during the light reaction. Then second is photophosphorylation. If we observe the definition. Photo means light. Phosphorylation means synthesis of ATP. Photo means light. Phosphorylation means synthesis of ATP. So here, ATP synthesis will be taking place in the presence of light. That's why it is defined as photophosphorylation. Phosphorylation means synthesis of ATP, and that is happening in the presence of photo means light. That's why it is called as photophosphorylation. And why it is needed during this light reaction? Just now only in previous slide. Yes, during this light reaction, we have discussed what this assimilatory power is utilized during the light reaction. What is assimilatory power? 3 ATP molecules with 1 NADPH. So where these molecules will be synthesized, that means during this light phase only ATP molecules and NADPH are synthesized. And that ATP synthesis is called as photophosphorylation, which is badly required during the process of non-cyclic photophosphorylation. And for non-cyclic photophosphorylation, uh, say this light reaction synthesizes ATP and NADPH. So synthesis of ATP from ADP and IP. IP means inorganic phosphate and ADP means adenosine diphosphate. That means formation of synthesis of ATP is possible by adding one adenosine diphosphate with one IP in presence of light within the chloroplast is called as photophosphorylation. And this photophosphorylation is of two types. One is non-cyclic photophosphorylation and the other is cyclic photophosphorylation. What is non-cyclic photophosphorylation and what is cyclic photophosphorylation? I'll discuss it in the coming slides. Yes. Light harvesting complex. Light harvesting complex. What does it mean? Several complexes are involved in this process, and the pigments are organized into two discrete photochemical light harvesting complexes. Those are number one, photosystem 1, the other one is photosystem 2. And these are named the sequence of their discovery. Here, these complexes, light harvesting complexes, LHC, they contain one. Here, a reaction center. Usually, reaction center is definitely it is chlorophyll pigment, either A or B. And surrounding this reaction center, there are other pigment molecules. Yesterday, we discussed about the accessory pigments. What are the accessory pigments? Carotenoids and phycobilins. Both are together called as accessory pigments. That means in addition to reaction center chlorophyll pigments, there are some other pigment molecules which surrounds the reaction center or called as accessory pigments. And this use of accessory pigments, what is the use of accessory pigments? This accessory pigments also helps in receiving the photon and they transfers it to the reaction center. As a result of that, the photons concentration will be increased in the reaction center. By that, reactions will be forwarded. That means this reaction center chlorophyll pigment by receiving more than enough photons, it excites and under that excited condition, it releases electrons which are furtherly accepted by primary electron acceptors. From there, reactions will be continued. That will be discussed in the other slides. Here, what we have to understand is light harvesting complex means what? That means light harvesting complex. It consists the main reaction center, which surrounds by the accessory pigments, which helps in receiving the photons and transferring it to the reaction center for the excitement and the releasing of electrons. That is the concept. And here, based on this photochemical light harvesting complexes, there are two systems. One is photosystem 1 and the other one is photosystem 2. Now, move to the next slide. Here also the same is explained. PS1, that means photosystem 1, pigment system 1, that receives the wavelength of 700 nanometers wavelength light, that receives P700 nanometers wavelength light, is absorbed by this pigment system 1, and PS2, that means pigment system 2, will be continued by absorbing P680 nanometers of wavelength light. So here we should remember this point. PS1, at PS1, this chlorophyll pigment and accessory pigments, they receive the P700 nanometers wavelength light, red light. And then next in PS2, it absorbs P680 nanometers of wavelength light. That means P680 nanometers wavelength light is absorbed by pigment system 2. Here, the yellow color structure, it is center reactional or reaction center. That is nothing but chlorophyll A. And it receives P700 nanometers or P680 nanometers of wavelength light. And the green color structures, all these are accessory pigments, which also helps in receiving the photons and transferring it to the reaction center. And the photons, they are transferred, or photons, for the photons, what is the source? Means sunlight is the source for photons. And this sunlight emits the sun rays, from that photons are absorbed by the accessory pigments. And the chlorophyll A, by the receiving of these photons, that emits that releases the electrons, which are absorbed by primary electron acceptor. That was shown here clearly. So here one statement. In PS1, the reaction center is chlorophyll A, which absorbs a wavelength of 700 nanometers of wavelength of red light. And PS2, it also absorbs the wavelength of P680 nanometers of wavelength light. The next. Yes, one important thing, that is Emerson enhancement effect and red drop. What is Emerson enhancement effect and what is a red drop? Emerson enhancement effect. First, we'll discuss about this. Emerson is a scientist, and his friends, they performed experiments, and they found the rate of photosynthesis at different wavelengths, and they measured photosynthesis as quantum yield. Emerson and his friends together performed experiments and stated that the rate of photosynthesis is different at different wavelengths. And the measured, that means the measuring units of photosynthesis is quantum yield. So here, quantum yield definition is, it is defined as the amount of oxygen released per quantum of light absorbed. There is a definition of quantum yield. That means photons are absorbed during the light phase. And those photons are units for photons. during the light phase, during the by photolysis, they releases oxygen. So this quantum yield defined as the amount of oxygen released by photolysis per quantum of light absorbed during the light phase is defined as quantum yield. And while performing this experiment, they found out a graph by putting the results on a graph paper. Here, look at this. Look at the graph. Here, on one side, quantum yield of photosynthesis is marked, and other side, wavelength in nanometers. Here, colors, which here colors are also be shown in this graph. So here, this wavelength starts from 400 to 700, ends with the 700 nanometers. So, which color is suitable for the photosynthesis? They observed it by doing the experiments. And in this, they first performed in their first experiment, which was performed by this Emerson and his co-workers. In this first experiment, they performed their experiment by exposing the plant to shorter wavelength. First, they exposed one plant to the shorter wavelength, and next longer wavelength, and noted the yield, quantum yield of that different wavelengths. That means initially they exposed the separately to different wavelengths, and they noted the quantum yields. Afterwards, they exposed the plant, another plant, to both the wavelengths. Exposed the plants to both the wavelengths. That means a shorter wavelength and a longer wavelength. That means 700 nanometers and 680 nanometers. Both the wavelengths are exposed on the same plant on a same time. At the same time, they exposed to the wavelengths of different the same plant, and noted the result also. So 3 experiments were conducted by this people. In first experiment, one plant is exposed to shorter wavelength. And second experiment, they exposed it to longer wavelength. And in third experiment, they exposed the plant to both the wavelengths and noted the results are quantum yields. And what they stated is when the plant is exposed to shorter wavelength, they noticed some value of quantum yield. And when it was when a plant is exposed to longer wavelength, that gives us extra quantum yield than the shorter wavelength. That means, for example, at P700, quantum yield is 10, and at 680 nanometers, quantum yield is 50. And when they expose the plant to both the wavelengths at a time simultaneously, rate of photosynthesis is found higher than the individual wavelengths exposure. Here, the point is individually exposed wavelengths gives different amounts of quantum yields. But when the plant is exposed to both the wavelengths at the same time simultaneously, that results that gives more quantum yield than the individual exposures. That is called as Emerson enhancement effect. Means individual exposure results in a little bit of a quantum yield, but when the plant is exposed to the both the wavelengths of photons or both the wavelengths of light, then that results in increasing the photosynthesis rate or quantum yield. So that is called as Emerson enhancement effect. And here in this graph, see here, initially the rate of quantum yield was normal up to 600. But after reaching 680, here it is very clear. After reaching this 680 nanometers, there is a sudden fall of quantum yield. After 680, there is a sudden decline of quantum yield. That is called as red drop. That means starting from 400 nanometers to up to 680, the rate of quantum yield is gradually increased up to 680 nanometers. It is increased, but after crossing the 680 nanometers, there is a sudden fall. There is a sudden dropping of a quantum yield, which is called as red drop. So a red drop is defined. There is a sudden decline in rate of photosynthesis after crossing 680 nanometers of red wavelength, which is called as red drop. So this is most important investigation which was made by Emerson and his co-workers or friends. So this is about the Emerson enhancement effect. The next, light reaction. Under light reaction process, there are two phases. One is non-cyclic photophosphorylation. What does it mean? Non-cyclic photophosphorylation. We know the photophosphorylation meaning. That means in the presence of light, phosphorylation means synthesis of ATP. ATP molecules are synthesized in the presence of light. That means photophosphorylation. Then what about this non-cyclic? What does it mean? That means whatever electrons are released from a reaction center, they passes on through various electron acceptors, and finally they will not come to the same released reaction center. They are received by another reaction center pigment. That's why it is called as non-cyclic photophosphorylation. So non-cyclic means released electrons from one reaction center passes through various electron carriers and reaches another pigment system, but not to the same reaction center. That means PS2. That's why it is called as non-cyclic photophosphorylation. ATP synthesis will be taking place by not receiving the electrons to the same reaction center. That's why it is non-cyclic photophosphorylation. And this non-cyclic is the main pathway. In this pathway, ATP and NADPH molecules are synthesized, and this process starts with pigment system 2. That means reaction center chlorophyll A absorbs 680 nanometers of wavelength. So in non-cyclic photophosphorylation, what reaction center will be the chlorophyll A, and which type of wavelengths are absorbed by this reaction center means 680 nanometers of wavelength, like red light is absorbed by this reaction center. Definitely, these points should be noted. The next step, just only explain why it is a non-cyclic. That means electrons released from the reaction center of PS2 passes through various electron carriers and reaches pigment system 2, but not to the same reaction center. That is PS2. That's why it is non-cyclic. And location. Where does this non-cyclic photophosphorylation takes place? This non-cyclic photophosphorylation takes place in thylakoids of chloroplast. We know the thylakoids. Yesterday in our previous class, it was explained. Thylakoids are plate-like structures which helps in holding the pigments. And those thylakoids are arranged in stacks, which are called as grana. So, non-cyclic photophosphorylation takes place in thylakoids of chloroplast, and this thylakoid membrane cytochrome complex, which helps in photolysis of water, is present on the inner membrane of the thylakoid. There are two important things. Not only this complex, one more NADP reductase enzyme also be there on the outer membrane of a thylakoid. So, two important points in location. Number one is this non-cyclic photophosphorylation takes place in thylakoids of chloroplast. And besides that, there are two important things. One is cytochrome complex, which is situated in inner membrane of the thylakoid, and RUBP reductase, NADP reductase on the outer membrane of the thylakoid. These two are important to remember. Right. The next, one more important thing, that is Z scheme. What is Z scheme? All the reactions, whatever they are taking place in non-cyclic, they goes in a shape of Z. That means initially they start from the bottom and they goes to the top level, and from there they goes down and again they come to topmost level. That is, originally it is in the shape of N, but that continues to the PS1. That's why the shape of this changes. It goes on in the shape of Z alphabet. That's why this process is also called as Z scheme, because of the changes that passes up and down and passes to PS1. So the other name for non-cyclic photophosphorylation is Z scheme. Why it has got this name means the changes, they starts from downwards and it goes up and comes down and goes up and next passes to PS1. That's why the shape of these changes are in the structure of Z alphabet. That's why it is called as Z scheme. All these are about the introduction of non-cyclic photophosphorylation. Moving to the next slide. Here is a non-cyclic photophosphorylation cycle. And first of all, introduce the abbreviations which will come in this cycle. While explaining this cycle, all these terminologies are used. So first one is PEA. What does that mean? That means primary electron acceptor. Then second is PQ. What is PQ? Plastopinone. And third one is CY. What is CY? Cytochrome. And what is PC? Plastocyanin. And what is FD? Pyridoxine. So these are the important abbreviations which comes while explaining this non-cyclic photophosphorylation. So you should remember all these abbreviations like PEA and PQ and CY and PC and FD. And a few more important points. This non-cyclic photophosphorylation takes place in thylakoid membrane, thylakoids, and during this non-cyclic photophosphorylation, important events are photolysis of water and synthesis of assimilatory power. NADP gets first one is photolysis of water, and the next is NADP gets reduced to NADPH2. Then ATP synthesis takes place. That means phosphorylation in the presence of light. That's why it is photophosphorylation. So these are the important events of a non-cyclic photophosphorylation. Here, in non-cyclic photophosphorylation, why we have given this title means the released electrons Why it is called as non-cyclic means release the electrons from the reaction center, which bears chlorophyll pigment A of pigment system 2 passes electrons through various electron carriers like now just now yes like this primary electron acceptors PQ plastophenone and plastocyanin and the cytochrome and pyridoxine. All these are the electron carriers. So through these electron carriers, electrons they passes through. And finally, they will reach the pigment system 1, but not to the pigment system 2. That's why this is called as non-cyclic. And during this movement of electrons, ATP synthesis will be taking place. That's why it is phosphorylation. And all these changes are taking place in the presence of sunlight. That's why it is called as photophosphorylation, non-cyclic photophosphorylation. So that is the meaning of the title. And in this non-cyclic photophosphorylation, why the photolysis of water takes place? Means here, look at the cycle here. Here, one H2O molecule that splits into H plus and OH minus. And again, OH minus that splits parallelly into oxygen and proton. This oxygen that comes out as a byproduct, and all these protons they are transferred to reduce NADP into NADPH. And electrons, these electrons are transferred to the reaction center to compensate the electron loss. Whatever electrons are lost by this reaction center in forwarding the non-cyclic photophosphorylation will be fulfilled by this electrons. That's why here photolysis of water takes place at most important event of a non-cyclic photophosphorylation. So electron loss in the reaction center of PS2 will be compensated by the photolysis of water, in which electrons are released and those electrons are transferred to the pigment system 2 of a non-cyclic photophosphorylation. And the changes in non-cyclic photophosphorylation will be explained in next classes. Okay. So today I will take leave here. Thank you. அந்த என்ன வேலை என்ன வேலை technical problem అని చెప్పాలి ఇది ఒకటే వచ్చి ఫ్రీగా ఇది ఆఫ్ అయినా కొట్టిమనేసి కింద కట్టడి చక్కెర ఇంత చక్కెర కొట్టిమనేసి దానికోసం చేసుకున్నట్టు ఇప్పుడు ప్లగ్ మీకు బాబు ఒరుడోరునా అయిపోయింది